So my name is Juliana Garcia, and today I'm going to talk about a paper-based detection of HIV drug resistance. HIV targets the immune system and weakens defense systems against infections and some cancers. HIV is transmitted via the exchange of a variety of body fluids from infected individuals, such as blood, breast milk, and other secretions. The most advanced stage is AIDS, and it can take from 2 to 15 years to develop. It has no cure and can be treated with ART drugs. Antimicrobial resistance, or AMR, occurs when microorganisms such as bacteria, fungi, viruses, and parasites change when they are exposed to antimicrobial drugs. AMR occurs naturally over time, usually through genetic changes. The medicines become ineffective and infections persist in the body, increasing the risk of spread to others. AMR threatens effective prevention and treatment caused by these microorganisms. HIV and drug resistance. In 2010, an estimated 7% of people starting antiretroviral therapy (ART) in developing countries had drug-resistant HIV. In developed countries, it was from 10 to 20%. Increasing levels of resistance has important economic implications as second and third-line regimens are 3 times and 18 times more expensive, respectively, than first-line drugs. The objective of this article was to see whether drug-resistant HIV paper-based detection can be made by oligonucleotide ligation assay or recombinase polymerase assay. Materials and methods. Synthetic DNA preparation. This one is the first step, so it is done to isolate nucleic acids from a patient's sample of blood. Synthetic G block stocks of the pol region are used to obtain template DNA for the assay. Two separate stocks are used. One encodes the sequence for wild-type HIV, and the other one encodes the sequence that include M184V drug-resistant mutation. So here we have a representation of the DNA preparation and amplification scheme. The blue arrows indicate the sections targeted by RPA primers for the integrated assay, and the green arrows indicate the sections targeted by PCR primers for DNA standard preparation. So these numbers you see here, underlined with a red color, are the mutant genes that can cause drug resistance. PCR. It is used to amplify DNA sequences from very low concentrations of it. It denatures, hybridizes, and extends a DNA sequence using heat, DNA polymerase, nucleotides, among others. It was used to determine concentrations standards. RPA. It is an isothermal alternative for PCR. By adding a reverse transcriptase enzyme to an RPA reaction, it can detect RNA as well as DNA without the need for a separate step to produce cDNA. It was used to amplify a sequence that contains several major drug resistance mutations in order to detect more than one in another assay. The oligonucleotide ligation assay. It is applied to discriminate between amplified wild-type and mutant sequences. It uses specific reporter probes for what wants to be identified. In this case, the reporter probes were used to distinguish between wild-type DNA and the mutant one, using FAM, deoxygenin, and biotin. So, here we have a wild-type DNA and a mutant-type DNA, and both are RPA products. So, the biotin will bind to both of the 3' extremes because they are the same in both types of DNA. But at the 5', we are going to have that in the wild type we're going to ligate deoxygenin, and in the mutant type we're going to ligate FAM. LFA and ELISA. So in this article they would use LFA and ELISA together so they can use it to confirm the presence or the absence of an analyte. There are plenty types of ELISA, but they all work with a similar structure. So they are going to use an antigen and an antibody. To the antibody, I'm going to bind an enzyme. That enzyme is going to react with its substrate and is going to generate a change of color only if there is a formation of the complex antigen and antibody. So I can see that change of color by spectrophotometry or a colorimeter. So here we have the RPA results. So in the first 2 lanes we have the DNA ladder that allow us to have these numbers right here that will allow us to value how many base pairs are going to be here. That in this case the RPA product is of 338 base pairs. So here we see that if we have no DNA, no amplicons are going to be seen. That if we have wild-type DNA and mutant-type DNA are going to have a similar amplicon line right here. That is a little bit gray because the concentration is low. But when we increase that concentration, the line is going to get darker and thicker. So here we can see a much thicker and darker line as we have more DNA concentration in both, in the wild type and in the mutant one. So here we have the oligonucleotide ligation assay results. Lanes number 1 and 2 correspond to DNA ladders that we explained before. So here in the lane number 4 we have the target of wild type DNA and the probe of wild type DNA too. So we hope to find a ligated product as we can see here because this too much. But here in the lane number 5 we have a wild type target and a mutant probe. So as data match, we cannot expect to see a ligated product here. So the same happens here in the lane number 6, but things change because the target would be a mutation DNA, but the probe will be a wild type DNA. So we cannot see the ligated product here neither. And here we in the lane number 7 we see the ligation product because there is a target that is a mutant DNA and the probe is mutant DNA too. So we hope to see the ligated product. And these two lanes number 9 and 10 correspond to a control of how would a wild type ligated product look like, and the same with the mutant one. So the LFA and ELISA results. We're going to have here the wild type detection zone and the increase of copies in the wild type DNA. So when we put more wild type DNA copies here, the wild type detection zone is going to increase its signal to background radio, and it is going to increase a little bit the mutant detection zone signal to background radio too. But when we see this one, that is for the mutant detection zone specifically, we're going to see that when we put more mutant DNA, it is going to increase in the mutant detection zone the signal to background radio, but the wild type detection zone is not going to increase its signal to background radio. The results depend on what we apply. So here in our target control, we must see no change in the color, and that's what we see. Here, when the 100% of the DNA is wild type, the wild type detection zone is going to detect it, so there is going to be a little change in the color. And here is the same, but with the mutant type DNA, as we have 100% of the mutant type. Here we see that the result depends on the concentration. So when we have nothing, we must not see a color change. When we have a little bit, we should see a little bit of color. And when we increase the concentration, we should see an increase in intensity of the color, and that is what we see here. The graphic shows that the signal to background radio is proportional to the amount of DNA of each detection zone. So this is what some authors said or proposed and was accomplished in the article. And so according to what Boyle said, it is a yes because each DNA strand is unique. According to what Pampertis said, it is true too because nobody would have to manipulate reagents when they're going to run the test. According to what Pampertis and Boyle said, yes, this experiment was based on a PCR and then mixed with OLA acid. And the Borman and Labar proposal were right when they said that amplification could be performed in paper because we saw that the RPA and the lateral flow assay and ELISA can make this thing possible. As a conclusion, we have that if this technique continues to develop and finds funding, it would be a very feasible way to know whether the patient's HIV is a resistant one or not, so we can redirect the treatment. And more alterations in the genome would be identified if the lateral flow test could be modified and some branches could be added to the ELISA design. So that was it. Thanks for watching.